In this video, we will discuss about this DNA cloning and genomic DNA library. In DNA cloning, we will discuss how the recombinant DNA cloning occurs. So first, we need to identify a, a part of DNA or the gene that we need to clone, and after that, if it is a recombinant, if we need two gene from two different organism needs to be combined, then it is called recombinant DNA. So, and people usually use different vectors in order to transfect the gene of interest or part of DNA into the organism, and then use the organism machinery to transcribe. So in this case, the donor DNA is the gene of interest, we can say, or the part of DNA which is of interest. So first, it's need to be integrated into the vector. Vectors are usually the plasmid or the small, you can say, small portion of small circular DNA that are usually found in the bacteria. So what initially does is, first the donor DNA is cleaved off with the help of some restriction enzymes, and the good kind of cleaving restriction enzymes are also known as molecular scissor, and it cleaves the DNA at a specific sequence of nucleotide. So and the good uh, cleavage is called as staggered cleavage. In this case, the one strand of the DNA is cleaved, so that during ligation step of donor DNA and vector, that is in this case, the ligation will occur in a more favorable manner, because the vectors and DNA are being cleaved by same restriction enzymes. So these that are nucleotides that are overhang nucleotides that are available in the donor DNA and the vector DNA are complement to each other, as it is shown here. This A is complement to T. So this TTAA is the overhang DNA of the vector, and AATT is the overhang DNA of the donor DNA. So during ligase step, this overhang strips will help the ligation step to go uh, much smoothly and without any trouble, and this will also increase the efficiency of the desired results. So what happens till now is that donor DNA is cleaved off with the restriction enzyme, and vector is also cleaved off with the same restriction enzymes, and they are mixed together. And while mixing, we also need to add the DNA ligase enzyme. What it will does is it will stabilize the phosphate backbone of the DNA, so that this ligation will remain stable. Otherwise, this ligation will not be stable and will get this donor DNA and vector will get disintegrate during the further step. Now, this is known as recombinant DNA, as part of the as vector is now being combined with the gene of interest. After that, this vector DNA then uh, we can say transcribe, or no, not transcribe. Uh, transfect this vector DNA, recombinant DNA, then we transfect into the bacterial cells, and these bacterial cells are being treated in a way that they can take the DNA, they can take the this recombinant DNA molecule. The membrane of the bacteria is more permeable to take the recombinant DNA molecules, and different combination. This is known as in within the bacteria, how the DNA are being present after the transfection of this recombinant DNA molecule. The recombinant DNA is also there, or bacterial chromosome is also there. So in this case, the final product will contain the bacterial chromosome translation and also the plasmid translation. So after this after separate we can say segregation of the product from the bacterial cells, we need to do further purification step in order to purify the uh, product that is formed from this donor DNA, and that needs to be and separation of this bacterial product that is formed from this bacterial chromosome translation. So this step is only till the formation of clones of this gene of interest, or we can say part of part of DNA. So this is how the different clones of the gene of interest is formed along with the bacterial or the uh, cell machinery that has been used to produce these clones. After this, purification step needs to be done. Some of the commercial purification kits are also available, such as midi prep or mini prep, that can used to purify. It can be used to purify uh, plasmid DNA from the bacterial DNA from the bacterial. It can be used to purify plasmid DNA from the bacterial DNA. Next step is the formation of genomic DNA library. So what happens in this case is that the genomic DNA is basically the this uh, DNA, or we can say recombinant DNA molecules. So and this needs to be what happens in this case is that uh, instead of binding the DNA ligase separately, what people does is they inject the donor DNA and vector separately within the cells, and allow the cells to allow the cells to do the DNA ligation and form the products. So in this case, two kind of possibilities can occur. Not too many kind of possibilities can occur. First one is that this recombinant DNA will bind to another recombinant DNA, or this vector will bind to another vector, and the third one is that this uh, desired binding, that is vector will bind to DNA. And so in the genomic DNA we will get we will get the final product the that contains the combination of all the three kind of possible uh, all the three kinds of possible combinations available. So as it is shown here is that plasmid vectors is there, and the genomic DNA. From the genomic DNA we have cleaved off the gene of interest, and this plasmid are also been cleaved off, and this plasmid is ligate with DNA, and during this step it is not shown in this case, but here also it is formed. During ligation step three combinations are possible. First, DNA is cleaved DNA will bind to another cleaved DNA, plasmid cleaved vector will bind to another cleaved vector, and third one is the uh, our interested our we can say uh, our interested combination, that is cleaved DNA will bind to cleaved vector, and this whole mixture will then inserted into the bacteria for cloning, or we can say for amplification of genomic DNA library, and then DNA isolation will be done, and this DNA will contains the all the three combinations, and from this we need to do further purification to purify our DNA of interest, or our our combination DNA of interest, that is cleaved DNA and this cleaved vector. So this is just the elaboration of this mixing step, we can say in simple words. So that's all for this video. I hope you have understood some part, something about DNA cloning and what is this genomic DNA library. Thank you for your time. If you need to understand more about this detail, more about these topics, you can go to the uh, reference that has been provided in the description. Thank you for your time again.